Hello students, in paper 13A, that is Cell and Molecular Biology of M.Sc Zoology semester 3rd, in the previous lectures you studied about the two important components of cytoskeleton of eukaryotic cells. These are the microtubules and the microfilaments. Today we will study the third important component of cytoskeleton, which is the intermediate filaments and its types. Intermediate filaments are one of the three types of cytoskeletal elements, and they are tough and durable protein fibers. Constructed like the woven ropes, they are typically between 8 to 10 nanometers in diameter, which is between the microfilaments and the microtubules. In most animal cells, the intermediate filaments form a basket around the nucleus and extend out in gentle curving arrays in the cell periphery. The intermediate filaments are particularly prominent where cells are subjected to mechanical stress. Various names have been attached to the intermediate filaments that have a basis in the cell type in which they are observed. Thus, the intermediate filaments in the epidermal cells are called the tonofilaments. In nerve cells, they are referred to as neurofilaments, and in the neuroglial cells, they are designated as the glial filaments. In cross section, the intermediate filaments have a tubular appearance. Each tubule appears to be made up of 4 or 5 protofilaments which are arranged parallelly. The intermediate filaments are composed of polypeptides of a surprisingly wide range of sizes. Now the types of intermediate filaments. They are very heterogeneous from the point of their biochemical properties, and on the basis of these properties and their morphological and localization, they are grouped into 4 main types of proteins. Types of intermediate filaments: type 1, type 2, type 3 and type 4. It is on the basis of their proteins. Let us start with type 1 intermediate filament proteins. They are found primarily in epithelial cells and include two subfamilies of keratin: the acidic keratin and the neutral or basic keratin. The keratin filaments are always heteropolymers formed from an equal number of subunits from each of these two keratin subfamilies. The keratins are the most complex class of IF proteins with at least 19 distinct forms in human epithelial and 8 more in the keratins of hair and nails. The type 2 IF proteins include 4 types of polypeptides: the vimentin, which is widely distributed in cells of mesenchymal origin, including the fibroblasts, the blood vessels, endothelial cells, and the white blood cells. Then the desmins, which are found in both the striated and the smooth muscle cells. Glial filaments, which occur in some type of glial cells, such as the astrocytes and Schwann cells in the nervous system. Now the fourth type, which is the cyanamide. This is a protein which is very heavy, 230,000 daltons, which is also present in the intermediate filaments of muscle together with the desmin and vimentin. Each of these IF proteins tend to assemble spontaneously in vitro to form the homopolymers and also assemble with the other type 2 IF proteins, which are the vimentin, desmin and glial filaments, to form copolymers and heteropolymers. Now the type 3 IF proteins, These IF proteins assemble into neurofilaments, which is a major cytoskeletal element in nerve axons and dendrites, and therefore they are called the neurofilament proteins. In vertebrates, type 3 IF consists of 3 distinct polypeptides and it is so called as a triplet. The type 4 IF proteins, they are the nuclear lamins which form highly organized two-dimensional sheets of filaments. These filaments rapidly disassemble and reassemble at specific stages of mitosis. Now this diagram or this chart will clearly show the characteristics of 4 types of intermediate filament proteins. This is the types of the intermediate filaments, the components of the polypeptides, their cellular function. The type 1, which consists of the acidic keratins and the neutral or the basic keratins, their distribution is they are found in the epithelial cells and epidermal derivatives such as hair and nail. And their proposed function is tissue strength and integrity. The type 2 IFs are the vimentin, the desmin, the glial filaments and the cyanmin. Their cellular location or their distribution is these cells are found in the mesenchymal region. They are present in the muscle cells, the glial cells, and the cyanmin is also present in the muscle cells. So their proposed function we can say is the sarcomere organization and integrity. The type 3 intermediate filaments, their distribution if we see they are present in the neurofilament proteins and their cellular location is the neurons and their proposed function is axon organization. The type 4 intermediate filaments, their distribution is they are present in the nuclear lamins. Their cellular location is the nuclear lamina of the cells and their proposed function is nuclear structure and organization. So, today we studied the different types of intermediate filaments and their distribution. In the next lecture, we will study about their structure and functions.